A very good afternoon to everyone. So I hope I am live in the class. Welcome in the class. As we say in the starting, let's crack CSIR UGC NET together. Recently, a crash course is going on for GATE 2021, GATE 2021, and in the evening 5 p.m. every day, I try to teach a topic from the syllabus of GATE. So you're watching this video under the platform of Unacademy, and Unacademy is one of the India's largest platform for the preparation of CSIR UGC NET Examination. I'm your educator Farida Johar Bandukwala. I have teaching experience of 10 years, qualified MP SLET examination in year 2018. If you're opting for Unacademy, what all things you are going to get? You're going to get daily live classes so that you will be in the momentum of studying every day. Live classes is going to give you the liberty of asking your questions directly with the faculty members. Then the whole syllabus is very structured, that all the units of the syllabus is quite structured. Teachers, they try to take up those topics which are most important. Then live tests and quizzes are taken so that you can evaluate yourself that how much you have learned for so far, and unlimited access for all the videos and all the study material which has been recorded or which is to be published in that particular category. These free live classes are conducted on two platforms, that is YouTube, and it is conducted on special class. So YouTube classes you can attend directly by subscribing the channel and hitting the bell button for getting the notifications. You can just go in the live class and attend the class. And for special class, you need to download the Unacademy learning app. Download the Unacademy learning app and make your account over there. After that, you will be asked to fill the referral code. So fill my referral code, that is FP01, and by filling the referral code you will be directed towards the special videos. So you can watch these special videos of mine as well as other educators by using my referral code FP01. Then there is this plus subscription, and plus subscription is a paid subscription. For 12 months the amount is 10,395, and for 24 months it is 15,120 rupees. So that is after using my referral code and by getting a 10% discount. So do not forget to use my referral code for getting a 10% discount, so that it is going to cost you that it is going to cost you a very less amount, a deducted amount, by using my referral code FB01. So you would need my referral code FB01 for your plus subscription as well as special classes. Now the topic of today is modification enzymes needed for RDNA technology, or we can say enzymes needed for DNA manipulation. For DNA manipulation. Right. So we'll be talking about all those enzymes which are actually responsible for producing our DNA. How our DNA is produced, we would need certain kind of enzymes so that we can cut the desired gene, and then introduction of this desired gene into the vector. All those things can be done only with the help of enzymes. So enzymes plays the most important role for construction of a recombinant DNA. So first of all, we'll be talking about the enzymes in today's class. Then we'll be taking up the process of RDNA technology. So first of all, we would need the enzyme which is responsible for producing the copies. So first of all, we need polymerases. So the kind of polymerase that we need is or template dependent DNA polymerase. If a sequence is there, the same sequence should be copied. So that is called as template dependent DNA polymerase. So that is the first thing. Template dependent DNA polymerase. This is the first thing. So Let us talk about template dependent DNA polymerase. So these are the DNA polymerases enzyme that synthesize the new polynucleotide just complementary to an existing DNA or RNA. If this is the existing DNA or RNA and a strand is formed over it, it is the work of template dependent DNA polymerase. So different types of DNA polymerase are used in the gene manipulation, which are in the category of template dependent DNA polymerase. So this is the first category. In this first category, let us talk about DNA polymerase 1. DNA polymerase 1. This DNA polymerase 1 is also called as Kornberg polymerase. DNA polymerase 1. So this DNA polymerase 1 has got certain activities. As we know, there are activities of uh, extending the strand from the existing 3' to 5' of the template strand, and for the new strand 5' to 3'. See, if this is 3' to 5 dash, this would be 5 dash to 3 dash. The two strands of DNA, they run anti-parallel, this we know. So the DNA polymerase 1 has got property of 3 dash to 5 dash and 5 dash to 3 dash exonuclease activity, that is cutting, and 5 dash to 3 dash polymerase activity. So what two types of activities it has got? 3 dash to 5 dash and 5 dash to 3 dash exonuclease activity. So exonuclease, the word itself is telling nuclease means cutting, and if it is cutting from the ends, it is called as exo. If it is cutting from inside, it is called as endo. So this type of enzyme, DNA polymerase 1, it has got exonuclease activity. It can cut in both of the direction it can cut. And then there is this 5 dash to 3 dash polymerase activity. 5 dash to 3 dash polymerase activity. These two types of activities are there, right, for DNA polymerase 1. After DNA polymerase 1, let us talk about reverse transcriptase. So, reverse transcriptase. So reverse transcriptase, it has got one more name, that is RNA directed DNA polymerase. It is going to make DNA from an existing RNA strand. So let us write its name. It is RNA directed DNA polymerase. And it synthesizes DNA from RNA. If this is the RNA strand, so it is going to make DNA strand from the RNA strand. That is reverse transcriptase. So reverse transcriptase was discovered by Howard Temin. Discovered by Howard Temin. And he did this work in the University of Wisconsin. And there is another scientist who did the work independently, that is David Baltimore, at the same time. So it was discovered by Howard Temin and the same work was done by David Baltimore. So both of them are given the credit of discovering the reverse transcriptase. So both of them, they shared the Nobel, both of them shared a Nobel for this, shared Nobel Prize for this in year 1975. And they got this Nobel Prize in the field of physiology, medicine. So reverse transcriptase. After that, in the category of polymerase, let us talk about new Taq DNA polymerases. Taq DNA polymerases. Taq DNA polymerase. So This we have read in our previous classes as well when we were doing PCR. TAC is Thermus aquaticus DNA polymerase obtained from the Thermus aquaticus. So they are found in the hot springs at a very high temperature. These organisms, that these bacteria, Thermus aquaticus. So this bacteria has got a polymerase enzyme which can work at a very high temperature. So what's the need of this high temperature enzyme? When we do the PCR needs to be done in at various different temperatures, different cycles and different stages. So enzyme should be heat resistant. It should not be heat labile, that it gets degraded by heat. So if it's heat resistant, it is going to work well at a higher temperature. So that thing is done by TAC DNA polymerase. So it is derived from a thermostable bacterium. So this is a thermostable bacterium. So if this is a thermostable bacterium, its enzymes are also thermostable. Enzymes are also thermostable, right? So it operates at 72 degrees Celsius, operates at 72 degrees Celsius, and it is stable even above 90 degrees, stable above 90 degree Celsius. So this TAC DNA polymerase, it is used in PCR. It is used in PCR. Okay, so this was about the nucleases. So we have seen certain nucleases, C template dependent DNA polymerase we have seen. Then we have seen reverse transcriptase. Then we have seen TAC DNA polymerase. Now we will be talking about nucleases. Nucleases. So what is the work of nucleases? The term itself is clear. To break down the nucleotides. That is the work of nucleases. So the enzyme which is responsible for breaking down the DNA molecules by breaking the phosphodiester bonds that link the nucleotides together is nucleases. So what is the work of nuclease? Degrades DNA, breaks phosphodiester bond, breaks phosphodiester bond which link nucleotides together. So there are two different types of nucleases. We have seen one of the nucleases in DNA polymerase 1, that is exonuclease. So nucleases are again of two types, that is exonucleases and another one is endonucleases. So exonucleases is responsible for removal of nucleotides one at a time from the ends of DNA molecule. Removes nucleotides from ends of DNA. Endonucleases, they are responsible for breakdown of the DNA chain in the between, able to break the internal phosphodiester bonds. Breaks internal phosphodiester bond. So these are the two types of nucleases, the main major category. Now some examples of nucleases, specific examples, like these are the generalized thing. Now coming to the specific example. The first is mung bean nucleases. So this mung bean nucleases is a single stranded specific DNA and RNA exonuclease. Single stranded specific DNA and RNA endonuclease. So it's a single stranded specific. It is going to work on single stranded DNA or RNA, and it is going to cut the whole strand in between. So the word is endonuclease, mung bean nuclease. And where is it used, or what is it is obtained from? So as the word is saying, mung bean, so it is purified from mung bean sprouts, obtained from mung bean sprouts. So it digests these single stranded nucleic acids, but when the nucleic acids are in the double stranded form, it cannot work on it. So if there is a DNA molecule which is double stranded, it cannot work on it. Can't work on double stranded nucleic acids. Mung bean nucleases. Then second one, specific example for this is S1 nuclease. S1 nuclease. So S1 nuclease is actually obtained from rice. Obtained from rice fungus. Obtained from Aspergillus oryzae, the one which grows on rice. Obtained from Aspergillus oryzae. That is a fungus which grows on rice. So this enzyme is responsible for degrading the RNA or single stranded DNA. Degrades RNA or single stranded DNA, and it cannot degrade the double stranded DNA or RNA DNA hybrid in its native conformation. Can't degrade, like mung bean, can't degrade double stranded DNA, comma RNA DNA hybrid, just like our mung bean nucleus. Thus, its activity is similar to mung bean nuclease, but this enzyme will also cleave a strand which is opposite on the complementary strand. So that is the difference between mung bean and S1. The enzyme will cleave a strand that is opposite or nick on the complementary strand. So that is the unique feature of S1 nuclease, which is different than the mung bean nuclease. The enzyme will also cleave a strand opposite a nick on the complementary strand. This is about S1 nuclease. Okay, so if there is a strand like this and there is a small cleave, so this S1 nuclease can work on this. If there is a small cleavage in the opposite strand, it can cut this strand as well, it can work on this. This was about S1 nuclease. Now we'll be talking about RNase A. RNase A. So RNase A is an endonuclease. And this it digests the single stranded RNA. Where does it digest the single stranded RNA? At 3' end of pyrimidine residues. RNase A digests the single stranded RNA at the 3' end of the pyrimidine residues. So it can digest somewhere over here. That is RNA A. After that, RNase H. RNase H is also endonuclease. also an endonuclease, and it digests the RNA strand of RNA DNA heteroduplex. Digests RNA strand of RNA DNA heteroduplex. Heteroduplex. So if this is RNA and this is DNA, so RNA's H, it is going to cut the RNA of such kind of the DNA. So not so many nicks. It can cut it somewhere over here because it's a endonuclease, right? It cannot cut the DNA, but it can cut the RNA. Okay, so RNA's H. After talking about the enzymes which are responsible for these all things, let us move towards the another category, which is a quite vast category, and that is restriction endonuclease. Restriction endonuclease. So the word itself is saying restriction and endonuclease. It's an endonuclease with certain kind of restrictions. So what kind of restrictions? What things? So it is a sequence specific endonuclease which can cut only at specific sequences. That is why the word given is restriction endonuclease. So restriction endonuclease is first of all found in bacteria. So it's a bacterial enzyme. It's a bacterial enzyme. The bacterial enzyme, then it is responsible to form the cut in the double stranded DNA. Cuts double stranded DNA by cleaving the phosphodiester bond. By cleaving phosphodiester bonds. Phosphodiester bonds at specific nucleotide sequences which are called as restriction sites. Cuts double-stranded DNA by cleaving the phosphodiester bonds at specific nucleotide sequences known as restriction sites. Restriction sites. These restriction enzymes or restriction endonucleases, as they are responsible for performing a specific kind of cut, that is also called as molecular scissors. Molecular scissors. Restriction endonucleases. So the term restriction comes from the fact that these enzymes restrict the entry of foreign DNA in the bacterial cell. If a foreign DNA comes in the bacterial cell, it should not create any kind of problems and difficulties in the bacterial cell. They are cut by the restriction endonucleases. Restriction enzymes, therefore, they are believed to have evolved to protect the bacteria from viral attack. It is believed they protect bacterial cell from viral attack, as virus particle is going to hijack the whole nucleotide machinery of the bacterial cell. It has to be very much active in controlling this or to resist this thing. So the word or the enzyme which was developed by nature is restriction endonuclease. So the existence of restriction enzyme was first of all postulated by W. Arbor, and he noticed that whenever viral DNA of bacteriophage it tries to enter the host bacterium, it is soon cut down into very small pieces, and that is why he theoretized the presence of restriction enzyme. So in year 1970, there is a scientist, that is Hamilton Smith, and his co-workers. They were successful in isolating the restriction enzyme from the bacterium Haemophilus influenzae strains. So this is the history of restriction endonuclease. There are two scientists to whom the credit goes, that is W. Arbor and second one is Hamilton Smith. Hamilton Smith. These two are the scientists to whom one can give the credit of restriction enzymes. After that, let us talk about some of the special characteristics of restriction enzymes. The special characteristics of restriction enzymes. So restriction enzymes, they have certain unique features which actually make them indispensable in recombinant DNA technology and and biotechnology. So these features are like this. First of all, the restriction site sequences recognized by most of the restriction enzymes are actually palindromes. So restriction sites are mostly palindromes. Restriction sites are mostly palindromes. Second is the restriction site is recognized irrespective of the DNA source. Restriction site can be recognized irrespective of DNA source. After that, most of the restriction enzymes are highly specialized to such an extent that they recognize only single restriction site. One restriction enzyme can recognize one restriction site. These are some of the special features of the restriction enzymes. Now talking about the nomenclature of restriction enzymes, that how are they named. So the name of the restriction enzymes are divided into 4 parts. So first of all, I'll give you the example and then we'll do ECOR1. Here the E stands for the genus, that is Escherichia. That is the genus. Then coli is the Co is for coli. Co is for coli, and that is the species of the bacteria from which it is isolated. R is for the strain, that is RY13, that is the strain. After that 1. This 1 is the first identified or the order of identification in the bacteria, because there can be many other restriction enzymes also. So many restriction enzymes from one of the bacteria. So that is the order of identification. Order of identification in bacterium. In bacterium. So these are some of the nomenclature part. So it will be made up of 4 parts. Now talking a little bit about cleavage. So cleavage can be in 2 forms, that is the blunt ends or overhanging ends. Overhanging ends. 2 types of cleavages can be done. So when we are saying blunt ends, it is very simple that there would be no hanging. So we'll see both of the things with the example so that it can be very, very clear without any kind of confusion. So let us first of all see the blunt ends, how they are going to look like, and then overhanging ends or sticky ends. Sticky ends. Sticky ends. So like this it is. G, A, T, B, E, C. So if this is going to be broken down, it can be broken down into GAT, CTA and ATC, PAG. So can you see any kind of hanging single, triple nucleotide? No, nothing is hanging. So this is a kind of blunt end. Now for the same thing, if we go for a sticky end, so let us draw the sequence for this. GAA, TT, C, C, T, T, A, A, G. So how it is going to look like? The sticky ends will look like this. This is how the sticky ends are going to look like. And this is how we have seen some of the restriction enzymes. The restriction enzymes itself are of three types, that is type 1, type 2 and type 3. So we'll be seeing we'll do the comparison of type 1, type 2 and type 3. So the type 1 is single multifunctional. It's a single multifunctional enzyme. A single enzyme with multiple functions. Single multifunctional enzyme. Type 2 has got separate endonuclease and methylase activity. Separate endonuclease and methylase activity. Type 3 has got separate enzyme with one common subunit. Separate enzyme with one common subunit. After that, in type 1, there are three different subunits. Three different subunits. In type 2, there are two identical subunits. In type 3, there are two different subunits. Then type 1 is going to need ATP and magnesium ions. Type 2 is going to need magnesium ion only, and type 3 is going to need ATP along with the magnesium ions. Right? So these are some of the categories of restriction enzymes. Now just a second, I'll just add the slides and we'll be talking about the further enzymes. Now the next enzyme that we are going to see is ligases. So the word itself is clear, that the one which is responsible for sticking. So ligases are also called as molecular glue. Ligases are called as molecular glue. So they are responsible for joining the nucleic acid molecules together. Join nucleic acid molecules together. So they can join both DNA by DNA ligase and RNA by RNA ligase. So DNA ligase is also there. and RNA ligases are also there. So DNA ligase is responsible to catalyze the formation of phosphodiester bond between the 5' phosphate of one strand and 3' hydroxyl group of another strand. So DNA ligase is going to form the ligase for phosphodiester bond of the phosphate of one strand and hydroxyl group of another strand. So DNA ligase is actually magnesium ion dependent. Let's just rub this. Magnesium dependent. So most of the ligases, they are actually obtained from bacteriophage T4, mostly derived from mostly derived from bacteriophage T4. So the function of basically ligase in nature, biological function other than what we actually um, use, or other than the recombinant DNA, its biological function, DNA ligase is responsible for repair of single-stranded breaks or nicks. That is the function of ligase. So blunt-ended ligases and sticky-ended ligation, both type of ligation can occur. So there are types of DNA ligases. So let us talk about the types of DNA ligases. DNA ligases. There are two families of DNA ligases. My class is going on. Just